Bio 1 students. In today's lesson, we're going to be discussing photosynthesis. This is Chapter 8 presentation. Start with the introduction, which is exactly what is it? So photosynthesis is the process by which green plants and some other organisms use sunlight to synthesize, which is makes foods from carbon dioxide and water. And one thing I want you to pay close attention here is breaking the word apart. Photosynthesis. This is photons or light synthesizing, or making the food. First, we're going to discuss how organisms sustain life. We have different types of organisms. First one being is a heterotroph, which must eat food or organic molecules from their environment to sustain life. This is bacteria, protists, fungi, animals. This is us. Okay. Next, we have autotroph, which this makes organic molecules from inorganic sources. And this would be uh, like a plant, using the photons from the sun to create. And then you have photoautotrophs, which use light, light as a source of energy to make organic molecules, plants, algae, and cyanobacteria. So a photoautotroph is a subdivision of an autotroph. What's important to know is photosynthesis powers the biosphere. It's what creates life on Earth. So first of all, a biosphere is a region on the surface of Earth and atmosphere where living organisms exist, largely driven by the photosynthetic power of photosynthetic plants, algae, and cyanobacteria. And this creates an energy cycle. So cells use organic molecules for energy, and plants replenish those molecules using photosynthesis. In the process, plants also produce oxygen. Better in this diagram, here we have photosynthesis. We have organic molecules plus oxygen giving glucose, plus oxygen giving us that cellular respiration that we went over last week. And then we have it releasing water and CO2. Then photosynthesis takes that up, and it's a cycle all the way through over and over and over again, giving us life. So to understand this more in depth, we'll start carbon dioxide goes into the leaf. As we know, plants breathe in carbon dioxide and they breathe out oxygen. So carbon dioxide and light go into the leaf. In the leaf, it makes sugar. And it releases oxygen and water. Complex diagram. We have water going into the system along with light, giving us a light-dependent reaction. And we have a waste product of oxygen. When it does this light-dependent reaction, it is creating ATP and NADPH. In the light-independent Calvin cycle, we're taking in carbon dioxide, we're giving out a product of sugar, and we're breaking those bonds apart, going back into light-dependent reactions. So this is why it's important to know this equation back and forth. If you notice, this is the reverse of cellular respiration. And if we break down the equation, it shows that we have carbon dioxide and water going into the system, and we get oxygen and sugar out of the system. So photosynthesis, energy within light is captured and used to synthesize these carbohydrates or sugars. And we have the formula which we went over. So CO2 is reduced, water is oxidized, and energy from light drives this inorganic reaction, a metabolic reaction absorbing the energy. So energy within that light is captured and used to synthesize glucose and other organic molecules. Nearly all living organisms rely on photosynthesis either directly or indirectly. And there's two stages of photosynthesis as shown in that diagram. The first one is light reactions, where light energy is absorbed by chlorophyll and converted to ATP and NADPH. And then you also have the Calvin cycle, where the ATP and the NADPH is used to drive the synthesis of the carbohydrates. The first to understand some of the terms that we just went over, we need to know what chloroplast is. So this is an organelle in plants and algae that carries out the photosynthesis. Okay, the green pigment inside the chloroplast is called chlorophyll. The majority of photosynthesis occurs internally in leaves in what's called the mesophyll, as seen here. The mesophyll cells must receive light, water, and carbon dioxide. Carbon dioxide enters and oxygen exits leaves through pores called stomata. Okay, so anatomy of that chloroplast. The outer and inner membrane are separated by an intermembrane space. A third membrane, the thylakoid membrane, contains pigment molecules. Okay, so that's where the green is found. This membrane forms things called thylakoids. Okay, they are enclosed by a thylakoid lumen. You then have granum, which are stacks of the thylakoids. Then you have a fluid-filled region between the thylakoid membrane and the inner membrane space, and this is the stroma. Okay, so make sure to go through this slide again and look and understand these terms before the next. About those two stages of photosynthesis. Okay, we have light reactions or light-dependent reactions, and we have the Calvin cycle or light-independent reactions. Okay, so with separating those two, the light reactions or light-dependent reactions use light energy. They take place in the thylakoid membranes, and they produce ATP and ADPH as well as oxygen. In the Calvin cycle or light-independent reactions, this occurs in the stroma. It uses ATP and NADPH to incorporate CO2 into the carbohydrate. Okay, okay, we're taking the carbon dioxide and the water into the system, and it's putting out oxygen and sugar. Okay, so we could actually see what's happening in photosynthesis in the equation like we could for cellular respiration. This is just another version of the two light stages together, so you can see it in just a different model. Okay, so continuing with those two stages, the light reactions at the thylakoid membrane, the NADP picks up an electron to become NADH. ADP combines with another phosphate. This produces the oxygen that goes out of the system. And from the Calvin cycle, the stroma uses carbon dioxide. ATP and NADPH are formed to make the carbohydrate. And that's how the systems work together. Actions that harness light energy. So during photosynthesis, energy in the form of light is transferred from the sun to a pigment molecule in the plant. Light is essential to support life on Earth. If we did not have life, there'd be no life. Light is a type of electromagnetic radiation, and it travels as waves. We have short to long wavelengths, which is the distance between the peaks in a wave pattern, and on the electromagnetic spectrum encompasses all possible wavelengths of electromagnetic radiation. As seen here, okay, it goes over what's visible, gamma rays to radio waves, and so forth. Light also behaves as particles called photons, which is where the photo of the photosynthesis comes from. These are massless particles traveling in a wave-like pattern and moving at the speed of light. Shorter wavelength radiation carries more energy per unit time than the longer wavelength radiation. Molecules can absorb the energy of visible light in a way that does not cause damage. So we have these different photosynthetic pigments that's gathering these photons. So the thylakoid membrane of the chloroplast is filled with these photosynthetic pigments that this can absorb all colors except green. There are different pigments and colors. We have chlorophylls and carotenoids. Chlorophylls are green, carotenoids are orange. Pigments also absorb sunlight energy and reflect others. So leaves are green because they absorb red and violet and reflect the green wavelength. Hopefully you remember this from our art classes. Okay, absorption boosts electrons to the higher levels, and the wavelength of light that a pigment absorbs depends on the amount of energy needed to boost an electron to a higher orbital. So having different pigments allows plants to absorb light at many different wavelengths. So after an electron absorbs the energy, it is in an excited state and usually unstable. This releases energy as heat or light, and these excited electrons of pigments can be transferred to other molecules or captured. Okay, so the photosystems of the light reactions. Breaking these apart, photosystems 1 and 2. So captured light energy can be transferred to other molecules to produce energy and intermediate molecules for cellular work, very similar to cellular respiration. Cycloid membranes of chloroplasts contain two distinct complexes of which light excites the pigment molecules. We have photosystem 1, which was discovered first, and then photosystem 2, which is actually the first step in photosynthesis. Okay, so once again, there's photosystem 1, which was discovered first, and then photosynthesis 2, which is actually the first step in photosynthesis. So starting off with that first step, we have photosystem 2 and electron transport chain. So the initial step in photosynthesis is light excites electrons in the pigment molecules within the light harvesting complex of the photosystem 2. Energy is transferred to P68O, a pigment molecule. This oxidizes water, generating O2, oxygen, and hydrogen ions. Electrons exit photosystem 2 and enter an electron transport chain. This energy is used to make the hydrogen ions for the electrochemical gradient. Photosystem 1, the primary role is to make NADPH. Light hits these light harvesting complex of the photosystem 1, and high energy electrons is removed from a P700 pigment molecule and transferred to a primary electron acceptor. Okay, this ferrodioxin then accepts two high energy electron and transfers electrons to the enzyme NADP reductase. The electrons transferred to NADP, which also accepts hydrogens to produce NADPH. Okay, we have the linear electron flow, so electrons move linearly from PS2, because it was the first step, to PS1, and that reduces NADP to NADPH. So starting with step 1, light excites electrons within the pigment molecules in the light harvesting complex of PS2, as seen here. The excited electrons move down to an electron transport chain to a more electronegative atom following the red arrow. This produces a hydrogen ion electrochemical gradient. In step 1b, the electrons are removed from water, then transferred to a pigment called P680. This process is what creates the oxygen and places additional hydrogen ions in the lumen. Step 2, electrons from PS2, which is the first step, eventually reach PS1, which happened to be discovered first. A second input of light boosts them to a very high energy level. They flow to the path shown by the red arrow. 3. Two high energy electrons and one hydrogen ion are transferred to NADP to make it NADPH. This removes the same hydrogen ion from the stroma. In step 4, the production of oxygen, the pumping of hydrogen ions across the thylakoid membrane, and the synthesis of NADPH all contribute to the formation of the hydrogen electrochemical gradient. This gradient is used to make ATP via an ATP synthase in the thylakoid membrane, just like it does in cellular respiration. Is the overall explanation that I just went through of photosystems 1 and 2, okay, explaining how they work together. Next is we're talking about the formation of ATP in the chloroplast. So ATP synthesis in the chloroplast is achieved by sorry, I can never pronounce that word correctly, mechanism called photophosphorylation, driven by the flow of hydrogen ions from the thylakoid lumen interstroma via the ATP synthase. Hydrogen ion gradient is generated 3 ways. Okay, hydrogen ions in the thylakoid lumen by the splitting of water, hydrogen by the electron transport chain pumping hydrogen ions into the lumen, and hydrogen in stroma from the formation of NADPH.
In bacterial photosynthesis, a single photosystem is involved. When an electron is energized by absorption of light, it is ejected from the photosystem reaction center. The electron then passes down through an electron transport system and finally back to the reaction center. The energy released during this electron transport is used to produce ATP. Since the excited electron returns to the reaction center, this mechanism for making ATP is called cyclic photophosphorylation. No reducing power needed for biosynthesis is generated in this process. Plants and cyanobacteria utilize 2 photosystems which work sequentially to produce both energy and reducing power. First, a photon of light ejects a high-energy electron from photosystem 2. The electron lost from photosystem 2 does not return to photosystem 2, but is replaced by an electron generated from the enzymatic splitting of water and the release of oxygen. The electron then travels from the excited reaction center of photosystem 2 down an electron transport chain and finally to the reaction center of photosystem 1. This electron transport system generates a proton motive force that is used to produce ATP. Since the excited electron does not return to photosystem 2, this mechanism for making ATP is called non-cyclic photophosphorylation. When photosystem 1 absorbs a photon of light, it ejects a high-energy electron, which is used to drive the formation of reducing power in the form of NADPH. The ejected electron is replaced by an electron from photosystem 2. In plants, photosynthesis occurs in specialized organelles called chloroplasts. The internal membranes of chloroplasts are organized into sacs called thylakoids. Surrounding the thylakoid membrane system is a semi-liquid substance called stroma. Photosynthetic pigments are clustered together to form photosystems. When a photon of light strikes the reaction center of photosystem 2, it excites an electron. 2 water molecules bind to an enzyme at the reaction center. This enzyme splits the water and uses the electrons from the water to replace the electrons removed from the reaction center. Oxygen is produced in this process. The primary electron acceptor for the light-energized electrons leaving photosystem 2 is plastoquinone. The reduced plastoquinone passes the excited electrons to a proton pump embedded in the membrane called the B6F complex. Arrival of the energetic electrons causes the B6F complex to pump protons from the stroma into the thylakoid space. thereby generating a proton gradient across the membrane. Because the thylakoid membrane is impermeable to protons, the protons in the stroma must pass through the channels provided by ATP synthase. As protons pass through, ADP is phosphorylated to ATP and released into the stroma. This process for making ATP is referred to as photophosphorylation. When photosystem 1 absorbs a photon of light, its reaction center passes high-energy electrons to ferredoxin. The enzyme NADP reductase then transfers the electrons to NADP to form NADPH. Electrons lost from photosystem 1 are replaced by electrons generated from photosystem 2. A small protein called plastocyanin, PC, then carries the electrons from the B6F complex to photosystem 1.
Next, we're going on to the Calvin cycle, which is those lyse carbohydrates via the Calvin cycle. Okay, the Calvin cycle is also known as the Calvin-Benson cycle. Carbon dioxide is incorporated into carbohydrates. This is the precursors to other organic molecules where the energy is stored. Okay, so think about it. This is why we eat carrots or vegetables, because of the sugars that's in them. This is how those sugars are created and how we get the energy we need. So this requires a massive input of energy. So for every 6 carbon dioxide molecules incorporated, we get 18 ATP and 12 NADPH must be used. Product is glycerol aldehyde 3-phosphate, or G3P. Glucose is later made from this G3P in a separate process. Calvin cycle for phase 1 is called carbon fixation. So carbon dioxide is imported into this RUBP using RUBP carboxylase oxygenase, also called Ribisco. This reaction product is a 6-carbon intermediate that splits into 2,3-phosphoglycerate molecules, or 3PG, very similar to glycolysis, if we remember. In phase 2, we have a reduction, okay, and that carbohydrate production. So ATP is used to convert this 3PG into a 3-bisphosphoglycerate, okay, or 1,3-BPG. NADPH electrons reduce it to glyceraldehyde 3-phosphate, or this G3P. Okay, so these 6 these 6 carbon dioxide molecules give us 12 G3P. Only 2 of those G3P molecules are used for carbohydrates. The other 10 molecules must be used for the regeneration of this RUBP, or Ribisco. In phase 3, we have the regeneration of this RUBP. So those 10 G3Ps are converted into 6 RUBPs using 6 ATP. The Calvin cycles begins by using carbon from an inorganic source and ends with organic molecules that will be used to make other molecules. Okay, so a little intense, okay, especially with the letters. Starting like we do with cellular respiration, we start with phase 1, which is that carbon fixation. So carbon dioxide is incorporated into an organic molecule via Ribisco. Okay, in phase 2, we have reduction and carbohydrate production. So ATP is used as the source of energy, and NADPH donates high energy electrons. 3, we have the regeneration of the Ribisco. So 2 G3Ps are used to make glucose, or the sugars. The remaining 10 G3Ps are needed to regenerate RUBP via several enzymes. ATP is required for RUBP regeneration. Okay, and here we have that whole Calvin cycle, okay, in detail for you to study and go through, okay, and basically follow the carbons. So variations in photosynthesis. Environmental conditions can influence both the efficiency and way this Calvin cycle works by light intensity, temperature, and the water availability.
The Calvin cycle is a series of reactions that results in conversion of carbon dioxide into the organic molecules needed to build new cells. It occurs in the stroma, which is the area of the chloroplast surrounding the thylakoid membrane. During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
C3 and C4 plants. We first have photorespiration, which Rubisco functions as a carboxylase enzyme. So RUBP plus carbon dioxide gives us 2,3-BGs. C3 plants make this 3-BG. 90% of the plants on Earth are these C3 plants. Rubisco can also be used as an oxygenase, so it adds oxygen to Rubisco, eventually releasing carbon dioxide. This is called photorespiration. So using that oxygen and liberating CO2 is very wasteful. More likely, this happens in hot and dry environments. This is favored when CO2 is low and oxygen is high. Plants. They involve a mechanism to minimize this photorespiration. So the C4 plants make oxaloacetate of the 4-carbon molecule in the first step of carbon fixation. Okay, they call this the Hatch-Slack pathway. Leaves have 1 to 2 cell layers of organization. And so in those mesophyll cells, carbon dioxide enters via the stromata, and 4-carbon compound formed BEP carboxylase does not promote phosphorylation. It's just in there. We'll see that on the next slide. And then we have this thing called a bundle sheath of cells, which a 4-carbon molecule is transferred that releases steady supply of carbon dioxide, minimizing the photorespiration. Okay, so to understand that a little bit better, okay, these here are the steps, okay, that happens in C4 plants. Go over the differences. So which is better, being a C3 plant or a C4 plant? It depends on the environment. Okay? In warm, dry climates, C4 plants conserve water and prevent photorespiration. In cooler climates, C3 plants use less energy to fix the carbon dioxide. So therefore, 90% of the plants on Earth are C3 plants. Things called CAM plants. So some C4 plants separate processes using time, okay, which this is where we get the CAM or CAM plant from. CAM plants open their stromata at night. So carbon dioxide enters and is converted to malates. Stromata close during the day to conserve the water. Oxaloacetate is converted to malate, and the malate is broken down into this carbon dioxide to drive the Calvin cycle during the day. Here's the difference between those two systems. Okay, we have the corn on the left and the cactus on the right. Okay, so this is using the C4 plant method, and this is using the CAM plant method.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of two sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the ocean.
As an overview and summary of photosynthesis, okay, we have 6 carbon dioxides plus 6 water molecules and light energy give us 1 sugar molecule and 6 oxygen molecules. Autotrophs store chemical energy and carbohydrate fuel molecules they build themselves. Most autotrophs make their food through photosynthesis using energy from the sun. Photosynthesis occurs in the chloroplast, an organelle specific to plant cells. The light reactions of photosynthesis occur in the thylakoid membranes of those chloroplasts. Electron carrier molecules are arranged in the electron transport chains that produce ATP and NADPH, which temporarily store that chemical energy. The light reactions capture energy from sunlight, which they can change to chemical energy that is stored in those molecules of NADPH and ATP. The light reactions also release oxygen gas as a waste product, which is what we breathe in. The reactions of the Calvin cycle add carbon from carbon dioxide in the atmosphere to a simple 5-carbon molecule called ribuscose. And the Calvin cycle reactions use chemical energy from NADPH and ATP that were produced in the light reactions. So the final product of that Calvin cycle is glucose. The end of the lecture. Good luck on your assignments and quizzes.